Hello everyone, my name is Sung-Yon Kim. I am an assistant professor at Seoul National University. What my lab studies is innate survival behaviors such as ingestion, thermoregulation, and aggression. How the brain is able to sense the specific stimuli that triggers or moderates these behaviors from both within and outside the body, and how the brain integrates and transforms the multiple streams of information to appropriate behavior outputs, such as eating, turning on a heater, or attack. Today, I'm going to tell you about our work on ingestion, specifically about the termination of ingestive behaviors by mechanical distension of the digestive tract. All meals come to an end, and every drinking comes to an end as well. How is this happening? This basic phenomenon has fascinated scientists since mid-19th century, which led to the idea that animals use negative sensory feedback signals arising from the digestive tract organs to monitor and limit ingestive behaviors. The first line of such feedback signals are generated from the oral and oropharyngeal cavities. In this one of the early experiments, Roland Bellows created an opening in the esophagus, or fistulated the esophagus, such that any ingested food or water drains from the animal before it can pass further. He observed that a thirsty dog would engage in sham drinking for a few or tens of minutes, pause the drinking, and then start drinking again. And this and later findings led to the hypothesis that oral and oropharyngeal signals cause temporary satiation of hunger and thirst. But the contribution of each specific sensory modality is poorly understood and is just beginning to be explored. The other two negative feedback signals stemming from the stomach and intestines have been pretty extensively characterized. In these classic studies, Miller and Tobin used a balloon surgically implanted inside the stomach to find that gastric distension suppresses both feeding and drinking. And later investigations, such as those by Phillips and Poley, using pyloric cuff and gastric infusion of liquids, have demonstrated that the suppression of appetite by gastric loading was correlated with the volume of distension, but independent from the nutrition content or osmolality of the solution. So far, the vast majority of the literature consistently pointed to a key role for gastric distension or gastric mechanosensation in the control of ingestion. This has also been clinically utilized in the recently approved gastric balloon therapy for treating obesity. The inflated balloon inside patient's stomach induces early feeling of satiety during a meal and helps losing weight. Signals originating from the intestine also reduces ingestive behaviors. In contrast to the mechanosensory nature of gastric feedback signals, intestinal signals are primarily known to be chemosensory. For example, this study has fairly clearly shown that intraduodenal infusion of fats robustly suppresses feeding, but the injection of saline does not have an effect. And further works led by Jerry Smith, Terry Poley, and many others have convincingly demonstrated that gastric satiation is volumetric and intestinal satiation is nutritive. So among these mechanosensory and chemosensory feedback signals arising from the digestive tract, mechanosensation in the stomach and potentially other organs as well would provide the information on the amount of intake regardless of the composition of ingestion. Despite its well-established key importance in the control of ingestion and its clinical importance, a specific population of neurons in the brain that encodes gastrointestinal distension has not yet been identified. And the gut-brain pathways signaling the gastrointestinal distension and the circuit mechanism by which the signal reduces appetite remains largely unclear. So we set out to address these questions. But where should we start? The digestive tract, the oral and oropharyngeal cavities, the esophagus, the stomach, and intestines. The walls of these organs are extensively innervated by cranial nerves 5, 7, 9, and 10, and also by spinal afferents. These nerves transmit sensory information to the NTS, or nucleus of the solitary tract, in a viscerotopic labeled line manner. as well as the trigeminal nucleus in the caudal hindbrain. Both of these nuclei strongly project to the parabrachial nucleus, which is a small pontine structure that broadly innervates many forebrain and midbrain areas that are implicated in feeding and drinking. This architecture makes the PB well suited to play a key role in mechanosensory monitoring and feedback control of ingestion. Therefore, we focused on the PB, and among the heterogeneous subregions and cell types within the PB, we sought to identify a genetic marker for the specific PB subpopulation that is naturally activated upon ingestion that encodes mechanosensory signals from the digestive tract, and that transmits appetite-suppressing signals. Towards this goal, we first examined the parabrachial neurons that are activated simply upon water ingestion. Specifically, we analyzed the expression of FOS, a surrogate marker of neural activation, after providing free water access to thirsty mice. And what we observed is that FOS-positive neurons were highly concentrated in the dorsolateral subnucleus in a remarkably localized manner throughout the anterior-posterior axis of the PB. And from the Allen-Braid atlas, we found that this particular subregion of the PB is marked by the expression of the prodynorphin gene. So this led us to hypothesize that PB prodynorphin neurons might be selectively activated upon ingestive behaviors. To test this hypothesis, we repeated the same FOS experiment with a fluorescent prodynorphin reporter mouse. And we found that most of drinking-induced FOS-positive neurons were prodynorphin positive, as quantified here. And likewise, most prodynorphin positive neurons were also FOS positive. So these data indicate that prodynorphin might be a useful genetic handle to target the PB subpopulation that is activated upon ingestion. And we also did some fish experiments to show that the prodynorphin positive neurons are essentially glutamatergic and are completely separate from CALCA positive neurons. We then assessed the real-time activity of these neurons using fiber photometry. As many of you know, fiber photometry is a way of recording bulk calcium activity from a defined neural population expressing GCAMs. As you can see, PB prodynorphin neurons are acutely activated upon water intake, and the robust responses are highly time-locked to individual drinking bouts. But are these neurons responsive just to water? It turns out that these neurons could also be activated by the intake of diverse fluids, including basic bicarbonate solution, liquid food, hypertonic saline, as well as liquid silicone oil. And these neurons were also significantly activated upon the consumption of hydrogel and chow food. To explain the difference in response amplitudes, we plotted the average response amplitude against intake rate and found a strong positive correlation. So these data suggested to us that PB prodynorphin neurons can monitor the ingestion of any types of solids or liquids using mechanosensory signals from the digestive tract. So why not try out direct mechanical stimulations to test out this? We found that PB prodynorphin neurons were strongly activated by touching the tongue. And these neurons were also robustly activated by inserting a gavage needle into the esophagus. And these neurons strongly responded to gastric balloon inflation as well. We also noted that maintaining balloon inflation for an extended duration revealed an adaptation-like responses. So PB prodynorphin neurons seem to encode instantaneous changes in volume of the digestive tract rather than the steady-state volume of the digestive tract. By contrast, PB prodynorphin neurons did not respond to the distension of the duodenum, nor to the distension of the proximal colon. So the activity of PB prodynorphin neurons encodes mechanosensation from the upper digestive tract parts, but not the lower digestive tract parts. These results align well with the previous notion that gastric satiation is volumetric and intestinal satiation is nutritive. We further conducted a variety of control experiments. First, the responses of PB prodynorphin neurons were not modulated by the taste, osmolality, and temperature of ingesta. So these neurons likely do not receive gustatory, osmosensory, or thermosensory information. And second, these neurons were unresponsive to the tactile stimulation applied to the dorsum or whisker, or the sensory detection of palatable peanut butter, or to conditioned tone predicting water delivery. So the activity of these neurons was also unrelated with locomotor activity or the anxiety level. And neither the baseline activity nor the responses of PB prodynorphin neurons were modulated by the third state. So these neurons, PB prodynorphin neurons, respond to water intake to the similar extent, regardless whether the mouse is thirsty or not. To summarize the results so far, using fibrophotometry, we have shown that the PB prodynorphin population is activated by seemingly distinct stimuli, ranging from water and food intake to mechanical stimulation of the digestive tract parts. Then how are these stimuli represented by individual PB prodynorphin neurons? Are the same or different neurons respond to these stimuli? To address these questions, we turn to two-photon calcium imaging. For the sake of time, I won't go too deeply into the two-photon datasets, but here's the data that are directly relevant to the question. First, we found that more than 80% of individual PB prodynorphin neurons were significantly activated by water intake. The majority of the PB prodynorphin neurons responded to gastric distension as well. And most of the individual PB Pdyn neurons also strongly responded to oral delivery of a liquid drop. So here, a drop of water would not immediately reach the stomach, at least for 20 seconds. So here, we are assuming that this is a stimulation applied just to the oral cavity. And strikingly, registration of the neurons across these three experiments revealed that most PB prodynorphin neurons registered here, 52 neurons, responded to all three of these stimuli, 42 out of these 52 neurons, namely to water intake and gastric distension, as well as oral stimulation. So here we can conclude that most individual PB prodynorphin neurons represent integrated mechanosensory signals from distinct organs of the upper digestive tract. And because the same neurons are activated by either food ingestion or mechanical stimulation, these results strongly support the notion that PB prodynorphin neurons monitor ingestion using integrated mechanosensory signals arising from the digestive tract. We next studied how the PB prodynorphin neuron population receives mechanosensory signals from the digestive tract. To address this question, we first mapped the monosynaptic inputs of PB prodynorphin neurons using engineered rabies viruses. And the monosynaptic input neurons were found throughout the brain, but as we focused on the hindbrain regions to identify the sensory input, visceral sensory input regions, we noted dense labeling in the trigeminal nerve nuclei and several specific subregions of the NTS. And these are the specific regions that are known to receive sensory information from the mouth, the pharynx, and the stomach via cranial or nerve afferents. By allowing for additional synaptic spread of the rabies virus from the trigeminal nucleus or the NTS, we could further label the anatomically connected neurons in the cranial nerve ganglia, including the trigeminal, geniculate, and nodose ganglia, as shown here. So this image shown here shows a subpopulation of nodose ganglion neurons that project to the NTS neurons that are presynaptic to PB prodynorphin neurons. So these results, only some of them are shown here, demonstrate that PB prodynorphin neurons and cranial afferent pathways are anatomically connected with each other. And although I did not show it here, we also found that some spinal dorsal horn neurons also project to PB prodynorphin neurons. To determine whether the vagal or spinal afferents convey mechanosensory signals to PB prodynorphin neurons, We tested whether vagotomy or spinal transection abolishes the responses of PB prodynorphin neurons to gastric distension. And the results were very clear. The responses are fully abolished by subdiaphragmatic vagotomy, but the responses remained intact following the spinal transection. So, mechanosensory signals from the stomach reach PB prodynorphin neurons via the vagal, but not spinal pathways. And this is also consistent with the commonly held notion that physiological gastric distension is signaled by vagal afferents, whereas visceral pain signal is relayed by spinal afferents. So, PB prodynorphin neurons monitor ingestion using integrated mechanosensory signals from the upper digestive tract. And the vagus nerve conveys stomach distension signals to PB prodynorphin neurons. Now, what do they do when they're activated? To determine the causal function of PB prodynorphin neurons, we used chemogenetic and optogenetic tools. First, chemogenetic stimulation of PB prodynorphin neurons potently suppressed food intake in food-deprived mice, and also suppressed water intake in water-deprived mice, as expected for the neurons signaling gastrointestinal distension. But what surprised us was the observation that these effects were mediated by the reduction in the number of bouts, but not the duration of bouts. And this is for both feeding and drinking. So if the mouse became satiated for hunger or thirst during a bout, the duration of bouts should have decreased because satiation would promote meal termination. But here, the bout duration was not affected, indicating that activating PB prodynorphin neurons does not induce satiation. And the optogenetic stimulation of PB prodynorphin neurons also had the same effects. What's shown here is that we applied the laser stimulation only when the mouse licked the spout. So the stimulation was given only when the mouse already initiated drinking bouts. And this lick-paired stimulation of PB prodynorphin neurons again decreased the bout number, but not bout duration to decrease the water intake. So these results fairly clearly show that activating PB prodynorphin neurons suppresses the initiation of intake bouts and reduces bout numbers, rather than terminating already initiated bouts and decreasing bout duration. Additional real-time place preference tests revealed that activating PB prodynorphin neurons is avoided, and pre-stimulating PB prodynorphin neurons before the test session suppress water intake for at least 10 minutes, 600 seconds here. So we can infer that activating PB prodynorphin neurons produces a sustained appetite-suppressing signals. So while the activation of PB prodynorphin neurons is the strongest at the beginning of the intake, the appetite-suppressing effects are exerted over the next tens of minutes, thereby decreasing the bout number. And it is important to note that such decrease in bout number rather than bout duration is also observed when the stomach is distended. And next, we chemogenetically inhibited the PB prodynorphin neurons to assess their physiological roles. And this manipulation increased food intake in food-deprived mice, and also increased water intake in water-deprived mice, and also elicited a trend towards increased intake of hypertonic saline in salt-depleted mice. What's evident from these studies, these results, is that inhibiting PB prodynorphin neurons induces pathological overconsumption of a variety of food and drinks, but only for those that the mouse already has the appetite for. And this effect is reminiscent of the effects observed by the loss-of-function manipulation of many gut-borne biochemical signals. Together, our data demonstrate that PB prodynorphin neurons are required for the negative feedback control of ingestion. Then how does the PB prodynorphin population suppress drinking behavior? To get some clues into this question, we examined their projection targets. This is a 2 mm thick sagittal tissue section of the brain that expresses UFP in the PB prodynorphin neurons. I'm not going to show you the details, but we further showed that these neurons densely innervate many forebrain and midbrain areas that are critical for intake behaviors. And we have also demonstrated that the projection from the PB prodynorphin neurons to the periventricular hypothalamus play a critical role. To briefly summarize today's talk, we have demonstrated a neural circuit mechanism centered around the hindbrain PB prodynorphin neurons for mechanosensory feedback control of ingestion. Upon the receipt of the mechanosensory signals from the digestive tract, with the signals from the stomach reaching these neurons via the vagus nerve, PB prodynorphin neurons produce aversive and sustained appetite-suppressing signals that discourage the initiation of feeding and drinking. And the projection from these neurons to the PBH may play the key role in appetite suppression. Like any important scientific discoveries, our study raises numerous further questions. First, what are the downstream circuit mechanisms for appetite regulation? Where does the hunger, thirst, and GI distension signals integrated? Can we find the circuit-specific drug targets to mimic gastrointestinal distension and reduce appetite? And what are the specific NTS, trigeminal nucleus, and cranial nerve ganglion neurons that signal digestive tract mechanosensation, like the cell type markers? And how does gut mechanosensation interact and potentially are integrated with chemosensory modalities? And what is the anatomy and functional significance of oral mechanosensation? So these are some of the questions that we are currently following up on this project. And in a clinical perspective, our findings shed light on the mechanisms that underlie gastric balloon therapy, gastric bypass surgery, and vagus nerve stimulation for the treatment of obesity and diabetes. In case of gastric bypass surgery, the distension of the remaining stomach after the operation is thought to be exaggerated upon ingestion. The function of specific circuits resolved in our study may represent potential targets for therapeutic strategies that aim to treat obesity and related metabolic disorders. Okay, with that, I would like to thank my students, especially Dong-Yoon, Yuryang, and Minyu, who did a wonderful job leading this project that I talked to you about today. And I also thank collaborators and funding. Thank you all for listening for the long time, and I would be excited to hear any of the questions that you might have. And so if you have any questions, please send me an email. Thank you so much.